All right, well, like everyone else, this is simply an amazing day, and I'm so pleased and privileged to be here speaking with you. And now we're going to mix it up a bit, and we're going to talk about the elephants. So let's see if I can get this working. I need to point this at someone. I'll point at Jonathan. Is it it's the green?
Press the green one.
I'm pressing. Look, I'll show you. I'm pressing. Ah, there, it's worked. Okay. All right, so we're going to talk about genetic risk for cancer, elephants and cancer resistance, and then the novel therapeutic potential of elephants. But before I start, I just want to go over a couple of things very quickly, and we touched on this yesterday. This is a slide that I put together with one of our fantastic genetic counselors, Wendy Coleman. We just did a back-of-the-envelope calculation. What are the carrier rates of different genetic syndromes? And if you added them all up in the US, how many people are living right now, this moment, at risk for cancer because of a genetic disease? And that came out to over 1.7 million people in the US. And that's probably a low estimate. And that was using the number 1 in 5,000 for Li-Fraumeni syndrome that we heard about. But what if it's not 1 in 5,000? What if it's actually 1 in 2,000, as some people say? That means there may be, out of 319 million people in the US of A, nearly 160,000 people with Li-Fraumeni syndrome. What if it's actually 1 in 1,000? Recent work by Michael Walsh and other colleagues looking at genetic risk in children. As a control group, they had something called the 1,000 Genome Project, which are supposed to be healthy individuals, 1,000 healthy individuals. One of those, 1 out of 1,000, actually had a deleterious p53 mutation. If the rate is really 1 out of 1,000, that means close to 320,000 people living with LFS. And as a clinician, and my other clinical colleagues know, we're seeing more and more patients referred to us because of panel testing that didn't even realize they had Li-Fraumeni syndrome. When we think about newborn blood screening, and we heard this earlier today about blood screening in Brazil, in the US we screen for some of these severe combined immunodeficiency syndromes. Those occur at 1 in a million. But we look for them because if we find that 1 in a million kid, we can affect outcome. PKU, a metabolic syndrome, occurs in 1 in 10,000 to 1 in 15,000. But Li-Fraumeni syndrome, 1 in 5,000. Now, I'm not suggesting we need to screen newborns, but I'm saying maybe it's something we should think about. And I think this is a discussion we're going to have as the future rolls on. And what if it's actually 1 in 1,000? And you'll hear tomorrow afternoon about all of the interventions that we now have to try to change the outcome of patients. So again, this is stuff we're thinking about in Utah, making models and trying to understand it. The other thing I wanted to briefly discuss before we get into the elephants is this concept of circulating tumor DNA. There are new genomic technologies out there. In Utah, we're putting together a study about can we use these? Is it possible? What's the feasibility of trying to detect tumors early? Like anything new, we don't know if it works, but we're not going to know unless we try. And this is something we've talked to David about, maybe trying to do a joint study. So we'll talk a little bit more about that tomorrow, but there are things coming. There even are things as crazy as dogs sniffing out cancer. So there's lots of things out there to discuss. Everything needs to be tested in a rigorous fashion. p53. This is not a group I need to spend a lot of time talking to about p53. But there are 4 points I want to make. 1, and you've heard many of these today, half of all human tumors will be missing functional p53 regardless of Li-Fraumeni syndrome. Function actually of p53 decreases in the normal healthy population as we age. And of course, you've heard today, as we age, the risk for cancer goes up, so there may be a connection there as well. Normally, we have 2 copies of p53, 1 from mom, 1 from dad, and in the general population, still half of us get cancer even with those 2, but if you only have 1 working copy, like many of the folks in the room here today, your risk for cancer goes up 90%. How does p53 work? You heard a beautiful, beautiful lecture this morning from our Israeli colleagues about p53. Yes, clap. Everyone can clap for Varda. She's worth it. Okay, so guardian of the genome, right? So p53 has many different functions, but its 2 main functions when it comes to cancer, again, as Varda explained, stop the cell from dividing so you can try to fix it, If you can't fix it, p53 helps in the coordination of apoptosis, which is cell death or cell suicide. Okay? When I talk to patients about p53, I, like many, talk about the superhero of the genome. I talk about a gene. I imagine it in its underpants with a cape flying behind its back. Wherever there's trouble, p53 is there, 2 copies to do its job. Now, of course, if we don't have 2 copies, either in a tumor that develops or with Li-Fraumeni syndrome, what happens, you can't stop the cell from dividing. You can't fix those mutations. You can't kill that cell, and those genetic mutations accumulate. And what's a cell that never stops dividing, that never dies and has genomic instability and mutations? That's cancer. So that's just sort of the primer. You already knew that, but now you're refreshed after the coffee break. That's why p53 is important. Now, for many years in our lab, we had been very focused on trying to understand who's getting more cancer and why, right? Patients that we take care of, families that we take care of, but also dogs. How many people here have a pet dog? How many people know what a dog is? You're not raising your hand, sir, but we'll talk about that at dinner, okay? Dogs have an extremely high rate of cancer. Dogs get cancer at 11 times the rate of people. It's actually due to genetics. We call them purebred dogs, but that's just a nice way of saying inbred dogs, right? We don't call inbred people purebred. That's another talk for a different conference, okay? But dogs get an awful lot of cancer, and this provides a really wonderful window to try to understand where does cancer come from and how can we intervene. And we had spent many years with that question. Who's getting more cancer and why? So how do we stop it? Well, it was a few years ago that we were at a conference and we learned about these elephants, these amazing elephants. Now, you don't have to raise your hand. I know all of you know what an elephant is, I hope. But here's the thing you might not know about elephants. Elephants rarely develop cancer. Now, that is particularly surprising because elephants are 100 times the size of people. Now, you heard this morning about stem cells and cell divisions and how that increases the risk for cancer. Elephants, it turns out, also live 50, 60, 70 years. That many cells dividing decade after decade after decade after decade, just by chance alone, all elephants should be dropping dead of cancer. Elephants should be getting cancer at 100 times the rate of people. Now, if you think about it, a baby elephant born after 22 months gestation, 22 months of being pregnant, something my wife tells me would not be a good thing. Born at 300 pounds, also something my wife tells me wouldn't be a good thing. Those baby elephants go from 300 pounds to 10,000 pounds, over 10,000 pounds in less than 10 years, putting on 3 pounds of cell mass per day. Also something my wife says wouldn't be a good thing. That many cell divisions growing so quickly, just by chance alone, all baby elephants should be dead before they reach the age of reproduction. But what would happen if all baby elephants were dying before they could reproduce? Elephants would go extinct. And extinction is not a good strategy for survival. So here we have this paradox. Elephants should be all developing cancer and dying from cancer because of how large they are and how long they live, but they never do. And so, they rarely do. It's not that they never do, they rarely develop cancer. Well, Dr. Maley, one of our collaborators, was curious about this and looked in the genome of the African elephant, at the DNA. And what he found was instead of 2 copies of the p53 gene, elephants have evolved, ready, wait for it, 40 copies of p53. And Dr. Maley explained that perhaps he didn't know for sure, but maybe, just maybe, that could be one of the reasons why elephants are rarely getting cancer. So I spoke with Dr. Maley and I explained that we take care of patients with Li-Fraumeni syndrome. We have a lab focused on Li-Fraumeni syndrome and cancer risk. What if somehow we could get some elephant blood and study that elephant blood in the elephant p53 right next to the human p53 and try to understand why don't these elephants get cancer? Why do they rarely get cancer? And how can we learn about that to help people? Well, he thought that was a great idea, and he said, if you can figure out how to get elephant blood, give me a call, right? So I had no idea how to get elephant blood, and I went home and I forgot about the whole thing. Until a few weeks later, I found myself at the zoo with my children, And I explained to them how amazing it was that elephants don't get cancer and they have extra copies of p53, and isn't that amazing, and let's go look at the elephants. And they said, yes, Dad, you're very weird, but we'll go look at the elephants with you. And we learned at the zoo that they actually draw blood from their elephants once a week to make sure that their elephants are healthy. It's part of their routine vet care. So we were able to establish a collaboration with the zoo, the local zoo in Utah, and study that elephant p53, which we're going to talk about in just a second. We also actually were fortunate enough to team up with the Ringling Brothers and Barnum Bailey Circus. And they told us that they have the largest herd of Asian elephants in the Western Hemisphere and wanted to know if we wanted to collaborate when they were in town. They also wanted to know if we wanted free tickets to the circus for our family, so of course it was a done deal. So working together with the zoo, with the circus, with an amazing group of collaborators in the lab and colleagues around the country, we were able to try to understand why don't these elephants get cancer. And we actually published it. We didn't publish in Newsweek, by the way. Someone else published the Newsweek story. But we published the JAMA story, right? And so we were really excited to see this publication in a clinical journal about elephant p53 and about Li-Fraumeni syndrome. Now, one of the things, and I know the families are waiting for the science. You want me to dig into those Western blots, and they're coming, so don't worry. But one thing I'll say is what's been really amazing about this entire experience is when this came out, it really captured the imagination, really, of the globe. There have been over 35,000 individual media reports published on this. In every single one, I try to mention Li-Fraumeni syndrome, right? So we've talked about Li-Fraumeni syndrome on Good Morning America, on the Today Show, on PBS NewsHour. We always try to get our patients featured in there. And so if there's one good thing that comes out of this besides publicity about elephants, is really a recognition and an understanding of Li-Fraumeni syndrome. And that's really been very, very important to us. Thanks. Really, you should be applauding for yourselves. Let's go on to the science, though, all right? Because this is where it gets really very cool. So what we found when we took these elephant cells and we compared them to human cells, and this is work done by Lisa Beglin in my lab. She's here. You can talk with her at dinner. But when we looked at the cells, what we expected to see, we took those cells, we zapped them with radiation, right, to induce DNA damage. We thought what we would see is that the elephant cells would repair like that because they had that extra copy of p53. It turns out the elephant cells repaired at about the same rate as the human cells, to which we said, oh, shoot. And again, I'm modifying. That's not exactly the words we used. So we got it all wrong. Why aren't these elephant cells repairing any faster? But when we looked carefully and when we remembered that p53 has more than one function, many functions, but one of them is apoptosis, what we noticed, and that's what those little arrows are pointing to, is that the elephant cells seem to be dying at a very, very high rate compared to the human cells when they got DNA damage. In fact, at almost twice the rate. These black bars over here, the tall ones, are the elephant cells compared to the human cells. So lots of cell death. In the same conditions, the elephant cells were very, very sensitive to any type of DNA damage. And in fact, this is my favorite figure from the paper that we put together. Over here on the bottom, these are patients. These are some of our patients, 10 different patients that we have, who we collected their blood, same experiment, we zapped them with radiation, and we measured how quickly do those cells die. Well, guess what? They weren't dying very much. And that's, again, part of the risk for cancer, because they kept growing with their mutations. Now remember, Li-Fraumeni, one functioning p53 gene, almost 100% lifetime risk of cancer, all right? Now, you double that, you take a healthy control, you double the p53, the cancer rate goes from close to 100% down to 50%. So adding just one copy of p53 brings down the lifetime rate and increases, almost doubles, the rate of cell death, right? Because the cells are responding. Not all of them, but some of them. And then you look at the elephant cells. Elephants have over 5 times as much apoptosis or cell death, we think, because of their extra copies of p53. Now, this became a sticking point with the reviewers. There was a lot, when you submit an article to a scientific journal, they review it and they write back and they tell you, here are 10 things we want you to change, 50 things, 70 things, so on, okay? And you have a discussion. And so they said, well, this is great. We believe that elephant cells are dying more quickly than human cells. You've convinced us of that, but how do you really know it's because of p53? Well, one of the things that we did is we looked to see that these genes, which are actually retrogenes, which are smaller versions of the wild type, the regular p53, the conventional p53, were actually being made from DNA into RNA. So we showed that, so check. Then we looked and we actually saw that the RNA was being made into protein. That's what these little dots are. These are measurements of protein. And when we put the p53 into human cells, we could see that they were being made into the elephant protein. Check. But how do we really know the p53 was being turned on and working? Well, one of the ways that we did it is we looked at some of these different genes that get turned on when p53 gets turned on and gets activated. One of those genes is p21. So we looked and we saw that just like in human cells that get exposed to DNA damage, the p21 went up in the human cells and almost identically went up in the elephant cells. And when we measured actually the protein levels, there was much, much more of the p21 in the elephant cells compared to the human cells. So that was very exciting. And so we published the story and we started thinking about elephants. Now, a lot of times the question came up, okay, well, this is great, but how is it going to help patients? All right? How is this really going to make a difference for patients with Li-Fraumeni syndrome or even patients with cancer? You can't take from an elephant, right? People would always ask when I give a talk, can you put the elephant gene into the people? No, that's science fiction. Right? You can't do it. It's a great thought. Maybe we can find some of these drugs, these compound screenings that you're talking about. There's no way we can do it. Now, one other thing that's very interesting is when you look at these elephants and you think about it, right? We've talked about p53, you know, it's been known about, it's been discovered for decades. We've heard about, 15, 20, 30 years of p53 research in the laboratory, trying to find the right drugs. You know, we spend hundreds of billions of dollars every year on research and development in the pipeline. And the way I think about it is these elephants have had 55 million years of evolution to figure out how to avoid cancer, right? If they didn't have these extra copies of p53, they couldn't grow up to be big to be elephants. So it took them 55 million years. So in a sense, the elephant has had 55 million years of research and development to figure out how to avoid cancer. That's pretty darn good, right? And it's also pretty hard to beat. But again, the issue came up with, well, how is this going to help people? Well, about a year ago, I was here. Varda, you know where that is? You've seen that view before. Yeah, it's in Haifa, right? It's beautiful. Well, I was in Haifa giving a talk, but I didn't know it. At the time, in the audience was Dr. Avi Schroeder. Varda knows Avi. A brilliant, brilliant chemical engineer. What Avi does is novel drug development. He works with nanotechnology, and his specialty is getting drugs into hard-to-reach places. A nanoparticle is less than 1/1000th the size of a human hair. It's made up of a liposome, and you load it up with whatever payload you want. I didn't realize it, but Avi was in the audience when I was speaking, and he said, I need to meet Dr. Schiffman to learn more about his elephant p53 so we can put it into his nanoparticle, so we can put it into our nanoparticles. Right? And then Avi spoke, and I said, holy cow, a holy elephant. I need to meet Dr. Schroeder to learn about his nanoparticles so we could put our elephant p53 in it. And we've been working for the past year, many, many sleepless nights and conference calls at all hours of the day and the night to try to figure out how do we work together between Israel and the US, team science, to really make an impact for patients. How do we get that elephant p53 into the nanoparticles? And again, it sounds like science fiction, but it's really not. There actually are quite a few nanoparticle drugs that are out there. These are just some examples of the nanoparticles targeting the tumors. They go exactly to the tumors where they need to be. They open up and they release their payloads. Here's an example from Avi's lab. This is a cancer cell, and these little green dots, those are the nanoparticles. And again, there are many drugs already on the market. Some of you may have already been treated with some of these drugs that have chemotherapy loaded into those nanoparticles. And now what we're working to do to try to understand is, can we load the elephant p53, and we believe we can, into the nanoparticle, and will it make an impact? Will it make a difference? Now, here is some preliminary data. This is a colon cancer cell, a human colon cancer cell, and what we've done is we've put in the elephant p53 gene into this human colon cancer. So think about this, right? These cells, for the first time ever, as far as I know, in the history of the universe, here's a human cancer cell with an elephant p53 gene inside it. And we stress out that cell, right? We expose it to a drug that causes DNA damage, and we see what happens. And we look at cells that have the elephant p53 gene turned on and cells that don't have the elephant p53 gene on. And what we found was, after 48 hours, there was almost twice as much apoptosis, or cell death, in that colon cancer cell that had the elephant p53. Now, I want to stress this is very, very preliminary. We still need to understand, how does this work? And we need to know, is it going to impact patients? Right? At the end of the day, the last thing I want to do as an oncologist, as someone who cares deeply for my patients, about my patients, I don't want to overpromise and underdeliver. So I want to be very, very clear. I'm not standing up here and saying, oh, we found the cure for cancer, this is great, or the cure for Li-Fraumeni syndrome. But what we are saying is that the elephants, we think the elephants have figured it out. Remember, they had 55 million year head start on us. So they know how to rarely develop cancer. It may not be all p53, it may be some other factors as well. But what we're trying to do now is learn from the elephants, learn from what nature's already figured out, and how do we apply this to people? How do we deliver this now to people? And this is what we're doing. We're working with many people in this room, hope to continue to work with many more after this meeting, to really understand, how is this elephant p53 working? We know it works, we're pretty confident it works, but we need to understand, what is the exact mechanism so that we can load up those nanoparticles and do it in the right way, so that no one here, hopefully, in the future will ever have to get cancer again. Now that's way in the future, and again, I come back to, I don't want to overpromise and underdeliver. But for now, hopefully you'll agree with me, you'll never look at an elephant the same way again. So in summary, cancer is a genetic disease. Elephants rarely develop cancer. And this nanotechnology is really an interesting approach, a novel approach. Enough nanoparticles in a teaspoon to hit every cell in the human body. Working with Dr. Schroeder, working with colleagues in the room, what I really want to stress is that success in Li-Fraumeni syndrome is going to be a team science, and it's going to take every one of us here. And so that's why I'm so thrilled to be with you today. Here's just a snapshot of some of the team. I do want to certainly mention Lisa again, who really did the bulk of the heavy lifting of these experiments. I also want to shout out to Dr. David Malkin, who really has been instrumental in encouraging me and mentoring me along the way. And the very last thing I want to show here is, this is Eric Peterson. Eric Peterson is one of the elephant keepers at the Hogle Zoo. You'll never meet a nicer guy than Eric Peterson, right? You with me? Also, never meet a bigger guy than Eric. He's huge. He's like an elephant, but it's okay. Well, you know, I don't know if this new drug that we're designing is going to work, or if it's going to save lives. But what I do know is, a few months ago, Eric called me up on the phone, and he was at the zoo. And he said, Josh, I have some lumps in my neck. Now, normally, I'm an elephant keeper, so I would ignore them until they took over my entire body, but you're always talking to me about Li-Fraumeni syndrome. It's all you ever talk about is Li-Fraumeni syndrome, and you've got me thinking about cancer. Do you think I should be worried? So I said, I'll be right there. So I came down, I felt his lymph nodes, and he was actually diagnosed with lymphoma. And he said to me, and we've discussed that, he said, you know, if we hadn't been working with the elephants, if it hadn't been for Li-Fraumeni syndrome, I would have never been diagnosed until it was way too late, and we caught it very early. So again, I don't know what's going to happen with the elephants and cancer and how many lives it's going to save, but at least for now, it saved Eric's life, and that's good enough for me. So thanks so much for your time.
We have a couple of minutes for some questions.
Hey Josh, Ryan from Nationwide.
Hi.
Hey, so have you ever wondered, so if elephants have 40 p53 isoforms, right?
Yep.
And if not insignificant portion of the mutations in p53 have dominant negative function.
Oh, sorry, my time is up. I have to go.
Then have you thought at all about how elephants might deal with what have to be
Sorry.
with these dominant negative mutations
Right.
that might occur randomly? And I
Right.
mean, you'd have to suspect that they have some way of dealing with dominant negative p53.
So this gets, I'm glad I put these extra slides up. So this gets to the question of how is it actually working? And we have a couple of different hypotheses. One of the things, again, we're working with David's group to strategize about the best experiments is figuring out what is the mechanism, right? Are the p53 retrogenes, are they actually working? Are they making tetramers? They look like they're getting phosphorylated, but it may be what we are calling the decoy model. So it may be that you need at least one allele of wild-type conventional p53 working to get the effect, because what we think is happening, if you look here, is all these extra retrogenes are being made, and they may be broken down. They may serve as decoys for miRNA, so that the MDM2 is breaking, well, that would actually not be that would be out in here, but the miRNA may be breaking it down while it's still a gene, while it's still being mRNA, but then when it gets out into the protein level, because again we showed it was made by protein, MDM2 may target it and sort of sop it up, and then leaving wild-type p53 to function. Now how that would interact with the dominant negative model, we're not sure. There could be enough of that extra p53 running around to quote-unquote distract it. So again, these are all great questions that we're trying to figure out.
Thank you for a very interesting talk. I would like to ask you, maybe I missed that. Do you put in nanoparticles the DNA, the genes, or the proteins?
No, well, this is why it's very exciting. So what we're working on is putting in the protein. So gene therapy is always a good thing to try, but we're not quite sure how we would regulate the gene expression and where would the vector land. So we think it's a safer approach to actually construct synthetically the elephant p53 and use that as the drug, use the protein. And then the next question that always comes up is, why elephant p53? Why not just use human p53?
Exactly, that was my second.
That's your second question. Very good, mind reader. Okay, so the reason is actually twofold. So the elephant p53s, they're shorter and they're more stable as a protein, so they're easier to transport, but also because of the way they're shaped, and again, we haven't figured out the exact mechanism yet, is remember they seem to shunt everything to apoptosis. So they work slightly different than the human p53.
You mean that you have elephant p53 without oligomerization domain?
Mm-hmm. Yeah.
Then how can it work as a transcription factor?
These are the answers we're trying to figure out now.
So have you tried to see, in reporters say that p53 can activate, elephant p53 can activate target genes, or how does it work?
Yeah, so we have RNA-seq data, and it looks like it's, what I should say is we haven't done the experiment yet of knocking out the elephant wild-type p53 and just replacing it with elephant p53 retrogene, but we know that the elephant cells, when they undergo DNA damage, that the whole p53 pathway is upregulated. So again, this is early times, but exciting times in trying to figure this out, because we want to understand the mechanism a little better so that when we go forward and make this drug, we know that it will work and we have an understanding of how it will work.
Good luck for you.
Oh, thank you.
Thank you, Josh. We really have to move on. We're running really quite late. Do catch him. He's not running anywhere.
That's true. Thanks.